you are about to hear represent one of the trademarks of Pepperdine University of Seaver College. While many colleges and universities now boast of undergraduate research opportunities in their curricula, at Seaver College it has been a part of our fabric of learning and discovery for decades. On our campus, students are co-creators of new knowledge. They are invited to contribute to every phase of research projects, whether it's an investigation on how drought impacts ferns, the filming of a new documentary, the study of student loneliness. Seaver College students are given the opportunity to get up from their desks, roll up their sleeves, and to learn by doing. Doing the field work, the data collection, the analysis, alongside faculty mentors. In fact, this event itself originates from undergraduate research. Madison, who we heard from this morning, a pre-med bio student, senior at Seaver College, one of the creators of today's symposium, discovered her passion for cancer research as a first-year student. During just her second semester on campus, she joined Professor Antonio Gomez's laboratory here at Seaver, studied the gene editing technology of CRISPR, and heard the call to study cancer biology. Since then, Madison has gone on to internships at the German Cancer Research Center and the Sarah Cannon Research Institute. Both of those experiences inspired her to launch this event. To all the students who are presenting here today, thank you for bringing your knowledge and your creative energy to our campus. Thank you for sharing with us your passion for learning and your pursuit of truth. Please remember, this is just the beginning of a really exciting scientific journey, the very start of your contribution to the world of medicine and science. We really look forward to hearing from you this afternoon. Please join me in welcoming our first student speaker, Seaver College senior Elizabeth Stansberry.
Well, hello everyone. Like Dean Kat said, my name is Elizabeth Stansberry. I am a senior biology major here at Pepperdine, and I'm so thrilled for the opportunity to get to share a little bit about my research at UT Southwestern Radiation Oncology Medical Center this past summer. I was an intern on a project investigating the dose interval effect in the spinal cord, specifically within the field of radiation oncology. So we begin with the question, why study the spine? And the answer comes down to this idea of metastasis that's been mentioned throughout the day today. Metastasis is when primary cancer in a certain cell tissue of the body spreads to a different tissue in the body. And that becomes a really big deal that we need to address. Bone is actually the second most common metastatic site for the 14 most common kinds of primary cancer. And so that means we really need to understand what happens when tumors develop in the spine and how we can address that. So this comes to the question of spinal tumors. So the spine and our spinal cord are responsible for really critical functions of the body. There's a reason why backbone is a metaphor for structural integrity, right? So when tumors develop in the spine, we have all sorts of things we're concerned about. We're concerned about primarily structural integrity. We can see vertebral body collapse when tumors develop in the vertebrae of the spine. We're also worried about threatening neuron protection. So we have these afferent and efferent neurons running in and out of the spinal cord, and when tumors develop in those hollows, we can see compression, pain, paralysis, restriction of sensation and function, and even the critical function of the spine protecting the spinal cord itself. When tumors develop in the spine, that they can press on the spinal cord, our highway of neural function in the body, and that can lead to pain and, again, paralysis. This is a really serious issue that we're looking at when we see tumors develop in the spine. How do we address that? Radiation therapy is a really effective tool for addressing metastatic cancer in the bone, and especially the bone of the spine. So it's important to understand that radiation therapy is typically prescribed with palliative intent. That is, it's intended to stop or slow the spread of the tumor, as well as reduce the symptoms that the patient's experiencing. And so we do see total or partial pain relief in the vast majority of cases. We can even more sensation and function from those tumors pressing from reducing the tumors that are pressing on the neurons. And we can even promote healthy ossification, that is, the healthy processes of bone building with radiation therapy. But how does that happen? I'm not a particle physicist, and I don't want to get too much into the weeds here, but it's important to understand the biological mechanism of radiation. So at its core, radiation is a delivery of energy, and that can be in the form of photons, protons, or even electrons, often by a machine called a linear accelerator. And if you've taken general chemistry, you know that absorbed energy breaks bonds. And that's our understanding of the mechanism of the damage that radiation causes, specifically breaking bonds within the DNA backbone. And this can happen via direct action, meaning that energy itself is breaking the bonds in the DNA backbone, or indirect action, where the energy is absorbed, creating free radicals, which in turn break the bonds of the DNA backbone. And so this is what we're doing when we're radiating the tumor in the spine or the spinal cord. But if you're tracking with me, you may have realized that we have a problem. When we're delivering radiation to patients, we're not only delivering radiation to the cancerous tissue, we're inevitably going to deliver some radiation to healthy tissue, and that's what we're calling the normal tissue issue, right? So treatment with radiation therapy becomes this optimization problem of how can we maximize the dosage that we deliver to the cancerous tissue while minimizing the damage that we cause to healthy tissue. And the field of radiation oncology has developed normal tissue dosage limits, which say how much radiation can we deliver to this patient before they reach this point of toxicity. And when we're talking about neural tissue, toxicity is when we deliver enough radiation to cause the death of oligodendrocytes. If it's been a while since you took physiology, you might need the reminder that oligodendrocytes are the cells that myelinate axons in the central nervous system, which means if we're killing those oligodendrocytes, we're going to cause a whole array of downstream issues. And so we want to avoid that by establishing normal tissue dosage limits that prevent the endpoint of toxicity. This is a critically important study to figure out what is the tolerance of normal tissue. And just to be clear, normal tissue tolerance studies don't just exist in radiation oncology. They exist for chemotherapies, for other drugs, and a lot of forms of treatment. So this is just one specific application of a normal tissue tolerance study. You might also be asking yourself, the radiation oncology field is not new. It's been around for about a century. Shouldn't we know by now how much radiation normal tissues can tolerate? Yes and no. So when the radiation oncology field first emerged, we would radiate wide swaths of the body, and in doing so, yes, cause damage to the tumors, but also cause damage to a great deal of normal tissue. And so, but as developments have occurred, we've been able to become more precise and more accurate with our delivery of radiation, and that in turn changes our understanding of normal tissue tolerance. So a couple examples of those developments. Image guidance. So we have MRI and X-ray image guidance that allow us to deliver radiation within a millimeter of certainty of where the tumor is, which is really exciting. Even now, today, patients can receive live tracking with imaging, meaning that as they breathe, the radiation site changes to track with their cycles of lung expansion, which is really incredible. Another change that has occurred within the past 20 years is the development of stereotactic body radiation therapy, which is a real revolution in the dose planning of the delivery of radiation therapy, which is in essence increase in the amount of radiation we deliver while decreasing the number of fractions or radiation sessions that a patient goes in for. And we've found that this can deliver the exact same amount of damage to tissues while decreasing the amount of toxicity that we see in normal tissue, which is really exciting. And then out of SBRT, we've also, in the past 5 or 10 years, there's been a development called pulsar, which is at its root ultrafractionated, meaning increased intervals in between radiation sessions, which again is decreasing the amount of toxicity that we see in normal tissue. So all of these different developments mean that we need adjusted dosage limits because normal tissue can tolerate more radiation when less normal tissue is getting radiated. A brief note on animal testing. So of course, human subjects are ideal, and we do use clinical trials and clinical data in these sorts of studies as well. However, when we're doing a study where we're looking to establish an upper limit, that means that the subjects in the highest dose are going to be receiving radiation that will cause them to develop paresis. And no physician would ever subject their patient to radiation that is expected or known to be too much radiation. And so in these kinds of studies, we do need to rely on animal